So, hi everyone, my name is Patricia Medina. I work in the Andalusian Center for Developmental Biology in Seville, Spain, and today I will talk about the functional annotation improvement of the regeneration process of a non-model organism using language models. We are working with Cleomdipteron, that is a novel non-model organism, to study regeneration, that belongs to the order of Hemeroptera, an ancestral group with most of the species extinct. This insect has a short life cycle of approximately 45 days that alternates the aquatic and aerial medium. Most of the time they live as a larvae in the water, they grow by molting several times, and when they reach a specific size, they emerge from the aquatic to the aerial medium as an adult, they mate between each other and start the cycle again. This insect has the capability of regenerate several of its organs in its aquatic phase in just 7 days. For example, in this image we can see how Cleomdipteron can regenerate the legs and the gills, that is the organ that we are studying and that the insect uses to move and to breathe for the aquatic medium in the larval stage, and they regenerate in just 7 days, as you can see in the slide. But they can regenerate as well the cercus, that is a sensorial organ that has at the end of the abdomen, and the antennas. This insect is really interesting because it's a fast regenerator. But we have some caveats in the regeneration study, mainly because it's a non-model organism with all the problems that are involved, and the regeneration itself remains poor understood, as well as the functional annotation of Cleom that is poorly characterized. For that reason, and in order to improve the functional annotation of Cleom, our first step was making a comparative genomics between Cleom and 21 invertebrate genomes in which its annotation remains good and present regeneration capabilities as well. As representative phylos, in this phylogenetic tree, we can find Arthropoda, in which we can find Cleomdipteron and Drosophila melanogaster. That is the model organism of reference that transfers the annotation to Cleomdipteron, and in which are based the main annotation file that we can find in UniProt associated to Cleom, with a total of 16,301 genes and 30,044 proteins. So, in order to find orthologs, we use the software SwiftOrtho to find orthologs in the next phylos: Metazoa, Placozoa, Nematoda, Chordata, Echinoderma, Cnidaria, Mollusca, and Arthropoda. And what we found is, if Cleom is a universe, that a substantial part of its protein is not covered with other species, and that it shares more group of homologs with basal animals than to the model organism of reference, that is Drosophila melanogaster, as you can see here in the circle. So, as a result of the analysis, how much can we map in groups of homologs? So, if we obtain a total of 48,398 group of homologs with all the organism, we can find 39,915 group of homologs that Cleom and Drosophila share with the rest of invertebrates, in which 17% and 16% belongs to Cleomdipteron and Drosophila melanogaster respectively, and the percentage sharing between each other is 9.8%. like a total of 6,506 genes that Cleomdipteron take from homology from Drosophila melanogaster. And how much can we map through Drosophila melanogaster? If Cleomdipteron has 16,301 genes, 11,942 genes are mappable to groups of homologs. From that number, 6,506 genes have at least a transferable by homology annotation to any Go category, that is transferred to Cleomdipteron, and 275 genes that we cannot transfer to anyone, and a specificity of 3,369 Cleom genes that are only exclusive for Cleom. So there is still a substantial part of Cleom that we cannot map anywhere. What we know about the annotation status of Cleomdipteron? If we check the annotation status of Cleomdipteron, we will consider the annotated proteins of UniProt as the gold standard, because we don't have any other annotation file of reference. So we are going to consider all annotation as the truth, because it's the only annotation reference that we have to compare with. So here we can find the mean number per protein of annotations that we obtain from UniProt or our gold standard in each category, in which some of them are taken from homology with Drosophila melanogaster, and most of them are automatic generated by UniProt algorithm. So the total number of annotations that we can find is almost 35,000 in 3 different categories associated to 9,927 genes. So almost half of the genes don't have annotations associated to our gold standard. So it is a lot. So in order to improve those annotations, we use some different predictors. First of all, we use Hammer, that is the most conservative predictor that searches domains in the protein sequence by hidden Markov models. In the second place, we use the DeepGo Plus, that uses the neural networks, sequence similarity, and protein-protein interactions to predict an associated function. As a novelty, we use Prot5 and SecVec, that are language models used in GoPretSyn, a method created by Burkhard Rost that basically measure distances between vectors, in which vectors are protein sequences embeddings. And here I attach the GitHub link if you are more interested and you want to find more about it. And this is what we obtained. You can see here the number of Go predictions that we obtain split by predictors and its categories. Although DeepGo Plus is the method that gives us more predictions, as you can see in mustard color, for biological process and cellular component, the language models predictors are very consistent between each category, in contrast to predictions by DeepGo, Hammer, and by orthologs that are super reduced. And here you can observe the number of proteins that we can annotate with every method. The consistency of the language models predictors in pink and blue colors is evident, and we can annotate most of the proteins with those, in contrast to the rest of the predictors. But which is the specificity of these Go annotations that we obtain with every method? Although we obtain a number of annotations, maybe those annotations are not relevant because don't give us so much information. For that purpose, we made the information content algorithm that basically gives us the specificity of the methods that is related to the probability of the occurrence of a particular Go term in a database of protein annotations. So those Go terms that occur less frequently will have a higher information content and are assumed to be more specific. So we can observe that although DeepGo Plus gives us so much predictions, those are not specific, whereas the language models, as well as the predictions obtained by orthologs that are transferred by homology and curated annotations, give us more specific predictions. So a good indicative of specificity for Prot5 and SecVec. So now, in order to assess the distance between the predictions obtained by every predictor and the gold standard, we collapse those predictions on guess categories. Here it is represented a Go tree graph in which we assess a hit, that is, if the prediction is exactly to the Go obtained by the gold standard, a close prediction if the prediction is a parent or a child from the Go obtained by the gold standard, and a related if the prediction is 2 or more distant from the Go obtained by the gold standard, and unrelated if the prediction is in a different branch according to the gold standard reference. So collapsing those Go's into categories, we obtain that the number of hit, close, and related predictions per category and method is bigger in the language models compared with the conservative methods that are Hammer and the searching of orthologs than DeepGo Plus and DeepGo, that most of them are unrelated. And here in percentage we represented the same. The number of predictions with specificity is bigger in the language models compared with the conservative methods than with DeepGo and DeepGo Plus. So in order to represent those numbers in number of genes with at least one prediction, we can extract that Prot5 is giving more genes annotated than SecVec and the rest of them. So the semantic similarity here gives us the relationship between a graph of code terms compared to the gold standard between categories. Most of the predictions obtained by categories are distributed in both sides of every plot, which means that half of predictions are related, but in the BPO, or biological process, plot, we can see that most of the predictions are related. So we determined that plot T5 annotate better in that category. So we selected plot T5 to annotate our genes and genes in regeneration. So to assess the regeneration process in our non-model organism, we focus on the study of the gills, an organ super interesting that they use to move and to breathe for the aquatic medium, and if you cut by the medial part or entirely, they can regenerate completely in just 7 days. So we set 3 different conditions in which we cut, wait for several hours, and extract completely the gills. Cutting after see a wound healing, after seeing early regeneration, and when the gill is totally recovered. And we compare those regenerating gills with the contralateral gills, in which we don't exercise any cut. We found that most of the differential expressed genes are in the first stage, in which the animals has to apply more metabolic effort than in the following 2, which it has sense. What we found mainly in the Richmond annotation of the first stage is an activation of the transcription factors mediated by GPCR receptors, an activation of immune system with a negative regulation of the humoral response, and active in the cellular immune response. Active metabolic process mediated by transmembrane receptors, amino acids and neuropeptides that mediates the nutrition and cell proliferation and migration, as well as apoptosis. So a lot of things related. In the second stage, we didn't find any enrichment annotation associated to cellular component, but we find a reduction of immune response that is related to inflammation and activation of receptors related with cellular sensing of the medium to answer with transcription factor activation in biological process and morphological function. And in the third stage, we don't find an enrichment annotation associated with morphological function and cellular component, but what we found in the biological process is a cellular response to external stimulus and to starvation with amino acid receptors activation in that category. So we can conclude that the number of functions are deactivating across each stage. So just to conclude, we identify the clustering of differential expressed genes that act along the process, taking into account the time, like a time course experiment. We did 2 different methods of clustering. By K means a hierarchical, and a cure for a optimization number of clusters, setting the number of clusters in 8 and using the hierarchical clustering. Once that we obtain those clusters, we reassign genes between the cluster 3 and cluster 6 and 7, because we see that follow a similar tendency. And to validate these functions with the regeneration process, we selected the cluster 8, in which before doing our input in the annotation, we don't know any functionality associated with them, and now, thanks to language models, we know. The group of functions that are activating and then deactivating and activated again a little bit are activation of apoptotic process, regulation of transcription, cell proliferation, phosphate and sialic acid transmembrane receptor, sensory perception of smell and taste, as well as feeding behavior, melanization defense, and synaptic activity. And to conclude, the summary is that language models enable to increase the functional notation in space in a reasonable manner, annotate better in biological process. We obtain acceptable level of main functional notations compared with classical predictors, and they can be used as an alternative in ORF genes. And as a future perspectives, we can develop a procedure to include annotations of all methods, and we have to validate the function of selecting genes involved in regeneration in organism in vivo. So thanks to the Casares lab for the experimental part, and Ana Rojas and the bioinformatics unit for the computational part, and thank you for everything.